Hello, everybody. My name is Choonbong Kwon. I'm protozoa taxonomist, especially ciliate, foraminifera, and rotaliid at the University of Ulsan, South Korea. Here, I would like to present my poster, Biodiversity of benthic rotaliid foraminifera in Dokdo. Thanks. First of all, I wanted to introduce our lab members. He is our professor, Professor Shin, Dr. Shajib, it's me, Lati, Sahal, Jamin, Nanda, Nobia, Lani, Lagib, Steve. This is our very nice colleague. Here is Dokdo. This study was carried out in a comprehensive inventory construction project to establish Dokdo biosovereignty in 2019. Among the protozoa, we studied foraminifera. In 1982, Dokdo was designated as a natural monument number 336, and Dokdo was management as a cultural property. Nowadays, it is important to secure biological resources in the world. Dokdo has relative poor terrestrial ecosystem species because it is a small islet and is affected by strong sea breeze that contain salt, and it is made of volcanic rocks of barren soil. On the other hand, the diversity of marine organisms is very high because it is located at the point where the cold and warm current intersect. Ocean currents are an important contributor to biota formation and change. So, this area is rich in species diversity. What is foraminifera? Traditionally, foraminifera is included in protozoa, but nowadays, foraminifera belong to SAR group. It belongs to a variety of marine environment. The foraminifera have a granular reticular pseudopodia, and they form a body with calcium carbonate as the main component, or mineral particle brought from the surrounding environment in the sea. The outer shell formed in this way can remain in the geological layer and become fossilized even after the foraminifera died. The morphological characteristics of foraminifera are as follows. Composition of the test, the structure, the number, the arrangement of inner chamber, and the shape and location of the aperture. Order Rotaliida is in the phylum foraminifera. Order Rotaliida has a typical enrolled multilocular test. The surface is smooth, papillate, costate, or straight, or cancellate. The aperture of rotaliid foraminifera is simple or with an internal tooth plate, intersolenium tube, or hemicylindrical structure. As a result, we investigated diversity of living benthic foraminifera in Dokdo. Among them, we are introducing now morphology of 13 species of rotaliid foraminifera. So, how we can collect foraminifera and take a picture and identify? Through scuba diving, about 1 liter of surface sediment at a depth of 3 to 30 meters was collected in a polyvinyl pack or like plastic bottle. The sediment sample were fixed by adding about 0.1 gram of rose bengal and 70% ethanol to distinguish between live and dead body. Sand sample were dry in an oven at about 60 degrees for 2 to several days. The dried sample was isolated under a dissecting microscope using a fine brush and a fine pull shape and identified using literature. Image of the front, back, and both lateral sides were taken of foraminifera selected as a conformation specimen using a Nikon SMG 18 dissecting microscope. In the case of very stereoscopic species, multiple high-resolution images were captured at different depths of focus and taken as a single extended depth of focus image. The slide specimen were attached to the standard permanent slide using a water-soluble adhesive for specimen preparation. So here we introduce rotaliid foraminifera, Begina cancris, Cibicidedos, Elpidium, Eponides, Planoglabratella, Neoconobrina, and Pararotalia, Globigerina, Ammonium beccari, Lobatula rosalina. This side foraminiferas from here, and there's foraminifera from here, and there's 3 species is common in both sides. Actually, to identify foraminifera, it is slightly difficult. Why? Many kinds of foraminifera was recorded in the fossil, but now we treated living foraminifera. So the recorded foraminifera, in the literature foraminifera, they describe or drawing the foraminifera is imperfect, the shell. Sometimes the shell was broken, but authors, they just drawing like present specimen, present cell. So, and also You can see the foraminifera, they have needs some crystal, like some color, like this. They have some special view in live specimen, but usually fossil foraminifera, they have no reveal like this, this character in like in the living specimen. So sometimes uh, we we are not easy to identify and find the same information about some species. So our about a conclusion about our result, uh, we treated living benthic rotaliid foraminifera, especially morphological diversity. So now we reported 13 common rotaliid foraminifera in Dokdo. For the work, we are planning to investigate taxonomically description and molecular characterization and the phylogenetic analysis of the foraminifera. Especially, we are thanks Industry Korea for collection of foraminifera by scuba diving and NIFS for allowing us to use the stereo microscope, so we can get like good high resolution pictures of our foraminifera result. Okay, thank you for listening. Say goodbye.